Good morning. We should start our seventh lecture on bioactive natural products. I wish all of you have sound health, and we are passing a terrible uh, and pandemic COVID-19 worldwide, and every day we are getting some bad news. We are expecting this bad time will be over. Anyway, I thank you all students. Uh, Amid of this pandemic, you are attending this Zoom lecture with limited internet facilities. Today, we shall discuss on um, phenolics or phenolic compound. You know, in our previous lectures, we discussed a lot about alkaloids and terpenoids compound. Phenolics are another third, largest group of uh, bioactive natural products, secondary metabolites found in plants and many other organisms. Phenolics are originated or derived from the biosynthesized from the Shikimic acid pathway. Now let us know how Shikimic acid are produced. Uh, Phosphoenol pyruvate from glycolysis, phosphoenol pyruvate is produced from the glycolysis process in the metabolic pathway, and a pentose phosphate pathway generates erythrose 4-phosphate. So these two, phosphoenol pyruvate and erythrose 4-phosphate, reacting together and develops shikimic acid, which are the precursor to phenylalanine and tyrosine. Phenylalanine and tyrosine, these two are amino acids. Phenylalanine lyase and tyrosine uh, ammonia lyase. Phenylalanine ammonia lyase and tyrosine ammonia lyase. These two lyases enzymes are involved in synthesis of cinnamic acids, coumarin, paracumaric acid, caffeic acid, ferulic acid, and sinapic acid. These acids are formed through the shikimic acid pathway. So these are the phenolic compounds. And from these phenolic compounds, lots of phenolic compounds are produced. Uh, Shikimic acid pathway is the main pathway for the synthesis of biosynthesis of phenolic compounds in the especially in the plant cell. Let us see the brief outline of the shikimic acid pathway. Here you can see pentose phosphate pathway and glycolysis pathway generates erythrose 4-phosphate and phosphoenol pyruvate. Phosphoenol pyruvate is the product of glycolysis pathway, and erythrose 4-phosphate is the product of pentose phosphate pathway. When they react together, they generate a new pathway called shikimic acid pathway. And shikimic acid pathway is involved in production of, you see, 4-hydroxycinnamic acid or paracumaric acid. And shikimic acid pathway also produce, as I mentioned, tyrosine, phenylalanine, and tyrosine then convert into 4-hydroxycinnamic acid or paracumaric acid. Similarly, shikimic acid pathway also generates phenylalanine. Phenylalanine, phenyl ammonia lyase enzyme activates and produce the cinnamic acids. Cinnamic acids to fumaric acid, paracumaric acid, ferulic acid, and different lignins are biosynthesized through this pathway. Similarly, flavonoids are generates from cinnamic acid pathway flavonoids, and different flavonoid polymer of flavonoids are called condensed tannin. So condensed tannin are also flavonoids, and they are the polyflavonoids. So cinnamic acid, two flavonoids, and then condensed tannin. Condensed tannin, found in many plants, even in the tea. On the other hand, shikimic acid pathway also generates tryptophan, and from the tryptophan alkaloids, this is an amino acid, indole 3 acetic acid, this is a phytohormone, very important phytohormone, and they are produced from the tryptophan. Similarly, shikimic acid pathway also generates protocatechoic acid, gallic acid, and gallic acid pathway finally generates hydrolyzable tannins. I mentioned another group of tannin, condensed tannins and hydrolyzable tannins. Hydrolyzable tannins contain gallic acid units. On the other hand, condensed tannin contains different flavonoids units. So this pathway, you must remember how phenolic compounds are biosynthesized in nature. So you can illustrate this pathway, very important. Here is the some example, no need to memorize, but if you carefully go through, you can see lots of phenolic acids, phenolic compounds are biosynthesized through the shikimic acid pathway. Phenolics, now question is, what are phenolics? Plants produce a variety of compounds that contain one or more phenol groups are called phenolics. So here you can see this is a benzene ring, and here which group is attached. This is the simple phenol. Phenolics are large group of diverse compounds. Many serves as defense compounds against herbivores and pathogens. Some are attractant of pollinator. In case of plant, plant flowers, they are colorful, and colorful substances are the phenolic compounds. Some absorb UV light and protect the plant from the damage of UVA from the sunlight. Some reduce growth of competitors. Some of the phenolic compounds have allelopathic effects to reduce the growth of other plants or other competitors. Phenolic compounds has some other functions. For example, they are very powerful antioxidants. So when we take phenolic, any plant materials, either fruits, vegetables, or any other, even grains, that contain phenolic compounds, they help us to keep strong immune by enhancing our immune system through the production of antioxidants, because phenolic compounds are antioxidants. They can neutralize the superoxides produced in our body. So one of the largest group of phenolic compounds are flavonoids. They are non-nutrients basically, but in 1936, Georgi thought that flavonoids are vitamin P. He proposed that Georgi Georgi proposed that flavonoids are vitamin P in 1936, but in 1950s, this theory was disproved. That means flavonoids have no activity of vitamin. It should not be considered vitamin P. That means the hypothesis is disproved. Late 70s, mutagenicity of quercetin was reported. Some scientists claim that quercetin, one of the important flavonoid, has the mutagenic activity. But recent research, it is confirmed that quercetin has powerful anti-carcinogenic effect. That means it can help us to flee from the cancer. that means anti-cancer principle are present there. It is called anti-carcinogenic. Carcinogenic means the compound possessing the cancer-inducing principle. It is opposite to the carcinogenic compound. Compound classes of flavonoids. There are different classes of flavonoids uh, in our natural system, and they have flavonoids have three rings. One is A ring, another is B ring. This is aromatic ring. This is aromatic, ring, a benzene ring, and C ring is not aromatic. Here you can see a bit three carbon ring. That means flavonoids C6, C3, and C6. So if you are asked briefly, flavonoid unit is C6, C3, 1, 2, 3 carbon here, C ring, and B ring 6 carbons present. So carbon 6, carbon 3, and carbon 6. So this is a simple structure of a flavone. It is called flavone, but it can be modified and produce various kinds of flavone. So this is one class of flavone, is a one class of uh, flavonoid. Another class of flavonoid is flavanone. Flavanone, how it is different from the flavone, here is the difference. Here there is no unsaturation. Uh, uh, this is the only difference. Here you see there is unsaturation, flavone. Flavanone, no unsaturation. Then flavanol. Flavanol means if flavone contains hydroxyl group in three carbon. Here three carbon if contains hydroxyl group, then it is called flavanol. Similarly, anthocyanidin. Anthocyanidin, here you see no substitution is here, almost similar to flavonol, but difference is here, no oxygenation is here. Flavanol, no oxygenation here, but it is different from flavanone. Flavanone does not contain hydroxyl group here, but flavanol, nol means contain hydroxyl group. So flavonol, flavanol, the difference is here flavonol contains oxygenation, here no oxygenation. Almost other things are similar. So this structure you need to memorize. You may be asked draw the structure of different classes of flavonoids with example, then you can write. Structure of flavonoids. Flavonoids I mentioned contains A ring and B ring, both aromatic, a benzene ring, and C ring like this. And this is the simple basic structure of flavonoids. Here, three this position is 3 carbon. Third carbon may contains hydroxyl. Similarly, generally in most of the flavonoids, these 5 carbon contain hydroxyl, 7 carbon contain hydroxyl group naturally, and here the 3 prime, 4 prime, 5 prime can be hydroxyl group or methoxy group and some other substitution. So flavonoids basic structure, this one can be substituted by different hydroxyl, methoxyl, or sugar moieties and can produce different compounds. Some flavonols, camphorol, quercetin, myricetin, they are very important. I mentioned they have powerful antioxidant activities and they are mostly carcinogenic. Here, camphorol is like this, simple flavonol. A simple flavonol, that means 3 carbon hydroxyl 5, 7, and here the only difference is quercetin and camphorol differences like that. You see camphorol, no hydroxyl group in the B ring of 5 prime. Then quercetin and myricetin contain one more hydroxyl group in the 3 prime. So myricetin contains 3 hydroxyl group, and then quercetin contains 2 hydroxyl group, and camphorol only 1. So these 3 structures are very easy to memorize: camphorol, quercetin, and myricetin. Two specific polyphenolics are elagic acids and isoflavonoids. Among the flavonoids, elagic acid and isoflavonoids, they are two specific flavonoids, very important and found in our many food materials. Elagic acid structure is like this. Elagic acid contains lots of hydroxyl group. Here. This is why powerful antioxidant and present in our tea. Green tea also contain elagic acid, catechin, and others. Elagic acid 4 arabinoside, that means elagic acid, one hydroxyl group can be substituted by an arabinose. That means sugar moiety can be easily connected to the elagic acid. Elagic acid 4 acetyl xyloside. Here arabinoside, here xyloside. Xylose, is acetyl group with sugar connected to the elagic acid. So these type of substitution are common in the nature. Lambar theanine, so elagic acid, how they can be also can be polymerized and form a different structure. Here it is shown, polymeric substances. Yes. Elagic acids also can be converted into elagic acid, elagitanin. This is called elagitanin, and obviously structural huge rearrangement taken place. Hydroxy, hexahydroxy diphenic acids. This is hexahydroxy diphenic acids. So 6 hydroxyl groups are present here, and here you see elagic acids contain 4 hydroxyl groups. Isoflavonoids share several features common with estradiol. Estradiol, this is a synthetic, compound. Synthetic, it can be synthesized, but naturally 17-beta estradiol is a female sex hormone. Isoflavonoid, natural compound. This is also naturally biosynthesized in human, but some plants also contain 17-beta estradiol. But if you see the structure of this one and isoflavonoids, there are some similarities. And interestingly, they also have similar, isoflavonoids have some estrogenic principle, show some female sex hormonal activity. Presence of a phenolic ring, prerequisite for binding to the estrogen receptor. The phenolic benzene ring can help them to bind, to the estrogen receptor in human. Pair of hydroxyl groups separated by a similar distance. Here you see hydroxyl group and this hydroxyl group and this hydroxyl group, this hydroxyl, quite similar distance. Common compounds, genistein or genistein or diargine. These are the some isoflavonoids. If you are asked the example of isoflavonoids that have estrogenic activity like genistein or diargine and diargine. And genistein and diargine, these two compounds are present in soybean. Soybean seeds contain this compound, and soybean seeds possesses estrogenic principle. Here you can see how structural similarities. This is why they both bind through the estrogen receptor. human estrogen receptor. Isoflavonoids, free and conjugated form, can be free and conjugated forms. Here, this is called eglycone. Eglycone means the basic skeleton of the isoflavonoid. We can call eglycone basic skeleton. And when it is, glycosylation called glycosides. So isoflavonoid glycosides, it can acetyl group can be added, or malonyl group can be added, and can produce more how to say, derivatives of the isoflavonoid. There are lots of phytochemicals contains antioxidant principles like carotenoids. We discussed a little earlier during terpenoidal compound, where mevalonic acid pathway produce the carotenoids. Pyruvate from glycolysis is decarboxylated to form acetate, which combines with coenzyme A to form acetyl-CoA. You know this one is acetyl-CoA. Here is the carotenoid biosynthesis pathway. No need to memorize, but you can see how they are biosynthesized. Nomenclature and structure, for example, beta carotene. Why beta carotene is very powerful antioxidant? Here you can see lots of unsaturation, double bond in this compound. And this unsaturation helps them to react with superoxide and neutralize them. Beta cryptoxanthin and lutein. Xanthin is very, very important, and beta cryptoxanthin also very important antioxidant. And commercially, xanthin 4 tablet you can find in the pharmacy. They are very powerful antioxidant compounds. So people who want to keep them free from stress, as well as those who are older than 50, they usually can take one capsule or tablet, capsule especially xanthin 4 available in the market, and very good for the health, especially to keep to boost up immunity. So structure-based antioxidant activity of the phytochemicals. Already I mentioned superoxides, different kinds of oxides, singlet oxygen or hydrogen peroxide. Different superoxides are produced in the cell due to stress or physiological activities, and they are toxic for the cellular system, metabolic pathway, and too much superoxides, if deposit in your body, then they will be harmful. Cell has its own mechanism to detoxify the superoxides, but if too much superoxide, then cell cannot detoxify it. In that case, we take lots of compounds through our food, especially plant materials. They contain antioxidant compounds, for example, phenolic compounds. They are antioxidant. That means can detoxify the superoxide and neutralize them, convert into water or like that, and then your cell can get it free from the attack of superoxide, because superoxide can damage the plasma membrane and can kill the cell, even can cause the cancer. So every day we need to take antioxidants from the plants, and they are called antioxidant phytochemicals. Here you can see lots of examples of antioxidant phytochemicals: para-hydroxybenzoic acid, para-fumaric acids. They have 0.08 antioxidant activity. This is some sort of unit, and from these structures, and here is the their power of antioxidant activity. What we can see among all these compounds, this one has the para-fumaric acid has the highest antioxidant activity compared to others. So there are lots of antioxidant compounds present in the plants, especially in the vegetables, fruits, as well as other plant materials, para-hydroxybenzoic acid, para-fumaric acid, protocatechuic acid, caffeic acid, vanillic acid, ferulic acid, syringic acid, synaptic acids. These are the antioxidant compounds, and all of them contains, you see, lots of hydroxyl group as well as carboxyl groups and unsaturation. This is why they can detoxify the superoxide. So structure of flavonoids already we discussed. The basic structure, A ring, B ring, and C ring, how they are and in the agglycone, this structure is present, and they can be modified through different enzymatic pathway and produce lots of compounds. Already I discussed this part. Here uh, uh, you see no number one. Number one is considered here, although not carbon. So in the numbering in the flavonoid ring starts from 2, from the C ring, 2 carbon, 3 carbon, 4 carbon, 5, 6, 7, 8, and then this B ring 1 prime, 5 prime, and 6 prime. As I mentioned previously, the tendency of hydroxylation as 5, 7, as well as 3 prime, 4 prime, and 5 prime is very easy, as well as 3 hydroxyl group is also possible. So quercetin is yellow, yellow color compound. This is a very famous, how to say, polyhydroxyl flavonoid. So it contains 1, 2, 3, 4, 5 hydroxyl group and a powerful anti-cancer, as well as powerful antioxidant compounds. Catechin is white. Here are no oxygenation, but all other structural, positions or structural diversity are same, quercetin and catechin. Catechin is white, and it is present in our green tea as well as the black tea. Catechin is very powerful antioxidant compound. So if you take tea regularly, you can find catechin there and can boost up your immunity. And cyanidin, also no hydroxylation here. Here is the double bond, here is double bond, is different, not double bond 2 to 3. Here you see 3 to 4 as well as 1 to 2. So this is the difference. Cyanidin. Cyanidin color is red, and many flower pigments are cyanidin. Here, quercetin 5-hydroxy group, it is shown here. Quercetin already we have shown, and catechin hydroxyl group is here, very clear, and cyanidin is different. So structurally similar compounds, but they have different antioxidant activity. So among quercetin, catechin, and cyanidin, which one is the highest? Which one poses the highest antioxidant activity? Quercetin. Antioxidant activity is 4.7 scale, and then cyanidin 4.4, and catechin 2.4. So they are powerful antioxidant. Importance of the 3-hydroxy group. 3-hydroxy group has some importance in antioxidant principle. Here you see same quercetin. If 3-hydroxy group are present, then antioxidant activity is 4.7. But if a glucose atom is connected to the glucose molecule is connected to the replace the hydroxyl group and produce quercetin 3 glycosides, then we can see the antioxidant activity decrease 2.5. Similarly, in luteolin, here you can see the difference is no 3-hydroxy group here, but structure is quite similar to quercetin. All are same except OH, 3 OH is missing. in luteolin, and its antioxidant activity is lower than the quercetin. That means 3-hydroxy group is very important, crucial for higher activity of antioxidant activity of flavonoid. Quercetin 4.7, antioxidant activity scale. But if we see taxifolin, contains 3-hydroxy, but no, you see, unsaturation here. This is why its antioxidant activity dropped only 1.9. So, 2 to 3 unsaturation plus 3-hydroxy is crucial for antioxidant activity. So here, summation, rutin, luteolin, these are different type of compound, quercetin, camphorol, taxifolin, they have different antioxidant activity depending on the, how to say, the substitution or connecting group in 3H or 3 carbon substitution, as well as unsaturation here is very important. Rutin also antioxidant compound, very famous, and rutin has higher antioxidant activity after quercetin, but other compounds have lower antioxidant activity. So this is all about today's lecture, and there are the textbook of bioactive secondary metabolites I shall share with you. I am not sure I have already shared or not. So you can get in the textbook, as well as I have shared already with you one very important chapter of the book on plant secondary metabolites in PDF format through your Facebook group. Please go through, and you can find lots of flavonoid compounds, their examples, their source, and their biological activity. That would be helpful for you. And please go through that PDF chapter, very, very comprehensive on secondary metabolites and in updated form. So this is all about today's lecture. I am expecting discussion from you or question, comments from your side. Please ask a question one by one. Please unmute yourself and then ask the question.
Sir, Assalamualaikum. This is Sharmin Sultana. Sir, I have a question.
So different colors are also especially colors of plants and other organisms. They are basically flavonoidal compounds. So even textile dyes, natural dyes in the fabric or clothes, they are originally people used flavonoid dyes. They are very good for the health compared to the synthetic dyes. So many countries and many peoples, they use the flavonoid dyes in the fabrics, and very good, good for health and safe for health. So coloring substances in the plants, especially fruits, vegetables, as well as in the flowers, all flower colors are flavonoidal compounds. Thank you very much. And they are polyflavonoidal compound, and they contain hydroxyl group and good antioxidant. They have the antioxidant principles. So they are very good for our health. Any more question? Next. Have you heard the lecture clearly?
Yes, sir.
Okay. Tofika, do you like to ask any question?
Sir, I have a question from previous class about terpenoid.
Yes.
As we know from our book and class that terpenoids are considered as secondary metabolites, then why we accept cis-terpene and diterpene as the example of gibberellin and abscisic acid? Because they take part in plant growth.
Why the gibberellic acid, abscisic acids, they are?
So they are secondary metabolites, but they are
Yes.
also taking part in plant growth.
Oh,
So
very good question. Yes, it is a very, very good question. You mentioned that some of the phytohormones like abscisic acids, gibberellic acids, cytokinins, they are terpenoidal compounds, so, and they are secondary metabolites, and they are involved in growth and development of the plants. Why they are treated as the secondary metabolites? Yes, this is a very, very good question. And it is an exception that most of the secondary metabolites, they are not directly involved in growth and development of the plant. But some of the phytohormones, they are involved in growth and development of the plants as the, hormonal substances. But if you carefully look at the abscisic acids as well as cytokinins, gibberellic acids, even indole acetic acids, they participate in or stimulate growth and development, but they never produce the energy, never produce the ATP. They never break down into water and or carbon dioxide, and the whole structure never used as the structural unit of the cell. Cell structure, cell contains cell wall, plasma membrane, and different organelles. They are not used as the structural unit, as well as never produce the energy, but they obviously stimulate the growth and development of the plant as hormone. So as hormone, they are the exception compared to other primary metabolites. Primary metabolites, they're directly used as energy source or structural unit. For example, proteins, they are the structure of the, a different organ. Our hairs or our teeth, they are composed of protein. Bones are composed of protein. But these hormonal substances, they are not the structural unit or energy source of the organism or of the cell or cell organelle. This is the difference, and this is a slight exception you can see. So phytohormones, although they are included in the terpenoids, they trigger the growth and development, but not directly used as raw materials or chemical substances as building blocks of the growth and development of the cell. Clear, Tofika?
Yes, sir.
Okay, thank you.
Thank you, sir.
Anyone? Do anyone have any other question? Very interesting question.
Sir, MD Saiful Islam, sir.
Yes, please.
Sir, most of the flavonoids has antioxidant activity.
Yes.
Here 4.7, 2.9 you have seen, but how can it measure?
Yes, there are some, bioassay method to estimate antioxidant activity of the flavonoids, depending on their hydroxyl group or unsaturation. There are some chemical reaction, DPP assay, it is called DPP assay. And there are some other bioassay methods to estimate the, power of antioxidant activity of any phytochemicals or any chemicals. So it is some scaling we can say. You can go, you can find the detailed methodology of how we can estimate the antioxidant activity of phenolic compound. And obviously, as I discussed in my lecture, that the presence of hydroxyl group as well as location of the hydroxyl group in the flavonoid structure and unsaturation, that is double bond in the certain location, they are critical and important for the higher antioxidant activity. Thank you very much.
Sir, is there any activity in agriculture?
Activity in agriculture, what does it mean?
Like as a growth and development of
Yes,
plants?
you know, not only human uh, uh, who suffers from superoxide. In plants, when they are in stressful environment, especially when they are exposed to salinity, drought, high temperature, and some other, or even infected by the microorganism, uh, pathogens or insects, they suffers from superoxide problems. That is, huge amount of superoxides are produced in their cell, and they are damaging for the plants. And you can see high salinity or a drought can kill the plants. The real thing happens through the superoxide activity can kill the cell and plants die. In that time, if plants have enough, antioxidants compounds, if they can biosynthesize, or even if you apply them in the plant exogenously from external sources, you can save the plant and can reduce their damage from the abiotic stresses. So plants, unlike like our human or other cellular organisms, they also have the superoxide problem, and they have their own mechanisms to detoxify the superoxides. Some variety you can see salinity tolerant. They have the higher ability to detoxify the superoxide generated due to the salinity problem. So it is genetically also control the mechanism, power of detoxification of superoxide, production or superoxide effect. And plants and all other eukaryotic cells contain cytochrome P450 system. There is another system in the cell, inbuilt system to detoxify them in our body also. So different enzymes, they are also antioxidant, superoxide dismutase or like that. So they can also detoxify the superoxide. So every moment, every second, cells are producing superoxides, and cell has the mechanism to detoxify them. But when you are uber stressed for some reason, for examination or some other stressful situation, or a lack of enough nutritionally balanced food, superoxide will produce in your, or carcinogenic food if you take, superoxide will generates and harm your cellular system. In that case, if you take phenolic compounds, or if you take flavonoidal compounds, you can keep you free from the damage of superoxides. You can keep your skin, very young, and the aging process accelerated due to the damage of the cells by the superoxides. So superoxides, are involved in making us or older, or relatively older, or like that. So aging process, becoming old, is also functions of the higher functions of the superoxide. So this is really critical for so dietary, yes.
Sir, do flavonoids have any antibacterial activity?
Yes, good
Or
question.
and sir, what about the dose, sir? If we take too much fruits or vegetable that contain flavonoids, does it do it will harm our health, or do will have any problem of our health?
Yes, you have two questions. One is whether flavonoids have any antimicrobial activity, and another question is if you take too much these antioxidant compounds as fruits and others, whether they are good or not. Yes, flavonoidal compounds, they have the antimicrobial principles. Some of them have very powerful antimicrobial compounds. So they are involved in defense against the microorganisms also. But all flavonoids are not powerful antimicrobial compounds, but some of them have a very powerful antimicrobial activity. Uh, Other question is if you take too much fruit or vegetables, whether good for health or not. Yes, one very important point is dose makes the poison. So flavonoid compounds, they have the antioxidant principles. You can take lavishly fruits and vegetables, but too much fruits and vegetables can upset your stomach and can be a big problem because it would be stressful for your digestive system. This is why we suggest to take, ideal amount, optimum amount of fruits and vegetables, but every day. Because if you take one day, it would not you would not get flavonoids after 3 days or like that. So every day, regularly, if you take optimum amount of fruits and vegetables, then it would be good for your health. And optimum amount of vegetables and fruits depending on the person, as well as person's age and other. So younger people can take a higher amount because they have the higher digestive capacity as well as metabolic activity, but older people, they have to be careful. And even younger people, any food, for example, mango contains lots of flavonoid compound, and mango, lychee, they contain lots of antioxidant compounds, good for our health, but take optimum amount that you can digest and you can tolerate, but you can take regularly. That is the advice. Thank you.
Thank you, sir.
Any question? Any other question? If you have no other question, I should say you thank you, and I hope you will stay safe and also with healthy life, and be careful about the pandemic COVID-19, and take much care to keep you free from any contamination. Thank you very much, and hope to see you tomorrow at 20 minutes past 10.